Good morning. So let's discuss some Viva questions. Like you see, these are basic questions. So define receptor is like one of toughest question. Okay. So it is basically a macromolecular protein. Okay. So this term is very important. Macromolecular protein. Okay. So where it is present and then how it acts, that is there in the definition. Okay. So basically, receptor is a macromolecular protein. Okay, now the drug and receptor bind to form drug receptor complex, and which after that forms a response. Okay, this is very important. Now there are two state models. So in first, receptor is active and the receptor is inactive. Now what we see, that this concept is like when receptor is inactive, an agonist, okay, agonist convert it into a receptor active, and an antagonist acts on active receptor and it inactivates the receptor. Okay, now what we see over here, that what is the concentration of RA versus concentration of RI, that is the active and inactive receptors. We see that an agonist has affinity, obviously they have an affinity and it makes the receptor active. The concentration of RA is more, right? In the partial agonist also, it also activates the receptor, but the amount of concentration of RA versus RI is lesser in comparison to the agonist. Now antagonist does not change anything. Antagonist does not change. Okay, and inverse agonist just reverses the concentration, that is it increases the RI concentration in active receptors than the RA. Okay, so these are the basic concept. Now there are many types of receptor. The first foremost is the ionic receptor, right? So you see the channel is, when the channel is closed, it's just like, actually this is a 3D model, but here we see this 2D photo. There are 2 alpha, 1 beta, gamma and delta. Okay, so now this is a closed channel which has an internal pore. Okay, and you see when there is a conformational change, and there are 2 acetylcholine receptors on the alpha particle, right? Now this channel is open. On the alpha subunit there are 2 active acetylcholine receptors, right? These are exposed by the conformational change. Now important is that there are many other receptors, and you have to know few examples of each type of receptors, because it will be there all over the book, right? So now G protein is the GPCR is the metabotropic receptors, right? So what happens when a receptor is there? Okay, now the GDP with alpha, beta and gamma subunit, that is in the G protein. So receptor with the G protein when acts, and this is the effector. Okay, so now the receptor and G protein when it's act, what happens? There is formation of this inactive substance. This one means first there will be receptor and GDP bound formation. What happens then? Then the GTP comes, and what will the GTP do? GTP will help the binding of the agonist, and it will replace GTP from its site. Okay, it will replace GTP from its site. So GTP will be there and GTP will be out, right? Now what will happen? Now the GTP, now what happens? The GTP with the alpha subunit leaves the beta and gamma subunit. Okay, and then it will bind to the effector. As you see over here, it will bind to the effector, and the beta and gamma receptor are what? They are separated. But now what happens? Now there is a hydrolysis of GTP to GDP. What again? There is alpha subunit. Alpha subunit has GTPase activity. GTPase activity, right? Now GTPase activity of alpha subunit removes the inorganic phosphate from the GTP and it forms the GDP, right? And the agonist from the receptor is thrown off. Okay, and again this alpha will bind to the beta, and this formation we see, this formation alpha, beta, gamma. Okay, this is the G protein. Okay, so this happens. All right, now G protein is nothing but a 7 membrane transmembrane helical structure. Okay, so G protein, you have seen the structure. This is the G protein. Now G protein is a 7 transmembrane helical structure which has GS, it may be GI or GQ, G12 or 13. What it means? It is G stimulatory, G inhibitory, GQ 12 or 13. Now this GS and GI, it activates or inhibits adenylate cyclase. It inhibits adenylate cyclase. Okay, and GQ, G12 to G13, it activates phospholipase C. Okay, now phospholipase C, what it does, it increases the IP3, DAG and calcium entry. Okay, as well as the most important thing is it acts on these 3 receptors, that is angiotensin 1 receptor, M1 receptor, alpha 1 adreno receptor. Okay, this is phospholipase C for you. Now adenylate cyclase GS, this is stimulatory type, is found in this receptors. Beta is adreno receptor, histamine receptor, serotonin receptor and dopamine D1 receptor, which increases the cyclic AMP. Now alpha 2 receptors, M2 receptors, opioid receptor, 5-HT receptor, there are some very few, and dopamine D2 receptor decreases the concentration of cyclic AMP because these are GI. So this is basic, this is which is what is GS and GI. Alright, okay. Now for example, we see there is a receptor and there is an agonist. Now it may be acting on GS or GI. So this is GS, this is GI, this is inhibitory one. Now what happens? It will act on the effector. It will act on the effector, and when it acts on the effector, this ATP will be converted into cyclic AMP. Okay, with the help of the enzyme adenyl cyclase. Okay, now cyclic AMP will convert into 5-AMP with the help of phosphodiesterase. Okay, now cyclic AMP will convert protein kinase into protein kinase in the active form. This is the phosphorylation where phosphodiesterase enzyme acts. Okay, now protein kinase will then release calcium, which increases even the lipolysis process. It increases the glycogen breakdown into glucose. Okay, and increases the function capacity of a heart muscle, and it decreases the glycogen synthesis. So it actually has a catabolic role. Okay, it breaks the glycogen, it breaks the lipids. Okay, with the help of even calcium release also there, right? So you should know about this. Okay, now this one, this is very important. Another example, this one GQ G12 13, right? Phospholipase C. Now you see over here what happens. A receptor agonist binds to the receptor. Okay, GQ receptor get activated. There is stimulation of PLC. Now what is PLC? PLC is phospholipase C. Now as the phospholipase C is stimulated, it then stimulates a very important molecule, that is PIP2. What is PIP2? It is phosphatidyl inositol 4,5-diphosphate. Phosphatidyl inositol 4,5-diphosphate. Okay, now in turn it stimulates DAG. So what is DAG? It is diacyl glycerol. It is diacyl glycerol, which in turn activates PKC. So PKC and PLC are different. What is PKC? PKC is phosphokinase C, protein kinase C. Okay, this is the protein kinase C. Now protein kinase C will then activate an ATP into ADP, and substrate will be turned into product. So this is the phosphorylation reaction. Another PIP2 will convert into IP3, that is inositol 1,4,5 triphosphate. IP3 will be converted to IP2, IP1, and then inositol and PI, and then PIP. So and then it will reconvert into PIP2. So in this process, what happens? IP3 will act on the calcium release, and calmodulin will act with calcium to convert E into calmodulin E, and there is this product as well as cam E will form a response. So this is a very complex process, okay, but it may be asked in Viva that how a receptor will act via phospholipase C route. Okay, so there are routes. You should know the routes. रास्ता पता होना चाहिए कि कौन सा रास्ता में चलने से क्या क्या मिलेगा. Okay, so there is a point A and point B, right? So तुमको यहाँ से लेके यहाँ तक जाना है. अब रास्ता ये भी है, एक रास्ता ये भी है, रास्ता ये भी है, रास्ता ये भी है. तो मतलब B इज़ द रिस्पॉन्स, ठीक है, विच यू वांट. ठीक है, वो किसी भी तरह से किसी भी रूट से आ सकता है. तो हमको प्रोमोटरी रिस्पॉन्स चाहिए, इनिबिटरी रिस्पॉन्स चाहिए, किस तरह का रिस्पॉन्स चाहिए, कैल्शियम के थ्रू रिस्पॉन्स चाहिए, उसका अलग अलग एक रूट है जो तुमको बार बार प्रैक्टिस करके जानना है. डोंट मेक इट सो कॉम्प्लेक्स, मेक इट वेरी ईजी. I've tried to make it very easy, but this is just a bookish thing. So you try your own concept, okay, and you make it very digestible, okay. अब देखो थर्ड इम्पॉर्टेंट क्या है, ये वाला देखो. दिस इज द टायरोसिन काइनेज बाइंडिंग डोमेन. दिस इज द टायरोसिन काइनेज बाइंडिंग डोमेन. दिस वन ये वाला, ठीक है. एंड दिस इज द एगोनिस्ट बाइंडिंग डोमेन, ओके. एंड दिस इज द एंजाइम इटसेल्फ. दिस इज द एंजाइम इटसेल्फ, ओके. नाउ व्हाट हैपेंस, देखो ये इंपॉर्टेंट है कि काइनेज का मतलब क्या होता है. काइनेज मतलब कोई भी हो जो फॉस्फोराइलेशन करवाए. काइनेज एंजाइम आर दोज एंजाइम विच डू द फॉस्फोराइलेशन, मतलब इनऑर्गेनिक फॉस्फेट को ऐड कराता है, ठीक है. ये इंपॉर्टेंट कंसेप्ट है. अब ये टायरोसिन काइनेज बाइंडिंग डोमेन जो तुम देखे हो, ठीक है, दीज आर सो इम्पॉर्टेंट. व्हाट विल डू दैट दिस होल सब यूनिट will be dimerized. So there is dimerization of the receptor and autophosphorylation of the tyrosine. So tyrosine जो amino acid है, tyrosine is an amino acid, yes, you have learned in biochemistry. Now tyrosine का autophosphorylation हो जाता है and there is a dimerization of the receptor. उससे क्या होता है, you see this kind of image is formed. What is this? This is tyrosine kinase phosphorylated. So there is autophosphorylation, okay, and there are two subunits which are joined together, right? Now there is a protein called GRB2. It binds to the tyrosine kinase phosphorylated form, and it will then, this enzyme complex will act on intracellular signaling protein, and it will convert into a cellular response, okay. What happens, that this cellular response is formed with the help of this enzyme complex. There are two enzymes in this, right? Now another thing, when we see over here, this is what tyrosine kinase, right? Now but when we see guanylyl cyclase, what it do, it converts GTP into cyclic GMP, and it will stimulate the cyclic GMP dependent kinase, and this again will it will help in the response in the cell, right? For example, there is a nitric oxide which acts via this route, cyclic GMP, okay. So you should know about this. This is very very important, right? Now few other few other like this is the last thing we are going to discuss. So what happens that we have seen there is dimerization, right? This is dimerization. Okay, now another dimerization I like to discuss. The first is heat shock protein 90 and 70 with immunophilin. Okay, this is one complex. Okay, and this is the receptor, and this is the globulin. Now what happens with the help of zinc fingers? With the help of zinc fingers, you see these are the zinc fingers in the receptors. What will happen? There will be homodimerization. So if there is a homodimerization, there is a heterodimerization also. So in homodimerization, what happens? The receptor, you see these receptors are what? This is the receptor with the with this globular subunit. It will form a product, okay, and which will act on the GRE element, okay, and there is the change, and there is transcription and mRNA, and then ribosome will help in the protein synthesis, and there is a cellular response. So okay, so this is just for a moment of dimerization. It will help in dimerization. Okay, so heat shock proteins, because when they will be joined, these two will be joined, so energy will be released, so this heat will be absorbed by this, so this hence it is known as heat shock protein. Okay, so this is important. Now something about agonist. Okay, something about agonist. This was all about receptors. Okay, now something about agonist. Now norepinephrine will act on alpha 1 adrenoreceptor, which stimulates to increase the concentration of IP3 and DAG. Angiotensin 2 will act on AT1 receptor, okay, and it will also stimulate IP3 and DAG, right, and these in turn activate the calcium channel, and there is a free calcium entry, okay, and there is some vasoconstriction. Okay, this is the process which we are seeing. ठीक है, लेकिन अल्फा 1 एंड्रोजन रिसेप्टर का ब्लॉकर कौन सा है? दिस इज द प्राजोसिन. ओके, एंड AT1 रिसेप्टर का ब्लॉकर कौन है? दिस आर द लोसार्टन. दिस इज द लोसार्टन. बट very important, very important, these two are competitive antagonist. But if we see, that is the second response. First response was this IP3 increase in DAG, and second response is this. Okay, so the first response, what will happens, this prazosin will compete with noradrenaline to produce an effect. Losartan will compete with angiotensin 2 to produce an effect. But here in this case, there is activation of calcium because of the increase of IP3 and DAG. Okay, now calcium channel blocker as such do not compete like nifedipine, do not compete with anyone, right? So it is a non-competitive antagonist. So the concept of competitive and non-competitive antagonist can be seen by this typical example. Okay, so what do you mean by competitive antagonist and what do you mean by non-competitive antagonist? Now for example, alpha 1 adrenoreceptor, okay, has a competitive antagonist of prazosin, as you have seen right now. It has a pseudo reversible antagonist called phenoxybenzamine, very important. It has an irreversible antagonist called dibenamine, and a non-competitive antagonist, as we have seen, that is the nifedipine. So it is very important in viva. This kind of questions will be asked. Okay, कि किसमें कौन सा रिसेप्टर किसका एंटागोनिस्ट है, कौन सा एगोनिस्ट है. ठीक है, फर्स्ट द डिफरेंस बिटवीन एगोनिस्ट, एंटागोनिस्ट, इन्वर्स एगोनिस्ट, पार्शियल एगोनिस्ट, जस्ट व्हाट वी हैव डिस्कस्ड. उसके बाद आएगा एक ड्रग जहाँ पे कौन सा रिसेप्टर पे कौन सा एगोनिस्ट है, कौन सा एंटागोनिस्ट है, इस तरह से कौन सा इन्वर्स एगोनिस्ट है. ठीक है। और दिस क्वेश्चन इज़ वेरी वेरी इंपॉर्टेंट कि अल्फा 1 एडिनो रिसेप्टर पे कौन सा कम्पिटिटिव है, कौन सा रिवर्सिबल एंटागोनिस्ट है, कौन सा नॉन कम्पिटिटिव एंटागोनिस्ट है, राइट, कौन सा स्यूडो रिवर्सिबल एंटागोनिस्ट है। दिस इज़ वेरी वेरी इंपॉर्टेंट। ओके, सो बेसिकली there is nothing much to discuss, just some basic concept of ANS. Okay, so when you have discuss about the receptors, थोड़ा सा ANS हल्का सा देखो. This is the peripheral nervous system. Okay, this is the peripheral nervous system. This is the autonomic nervous system and somatic nervous system. In the somatic nervous system, there is only a postganglionic nerve which acts on the skeletal muscle, and this is the NM receptor. Okay, in the neuromuscular junction. Okay, this is only important point about somatic nervous system. But in the autonomic nervous system, we have sympathetic and parasympathetic nerves, group of nerves, and in the sympathetic there is preganglionic nerve which will act on the adrenal medulla, on the NN receptor, and it will form noradrenaline and adrenaline. Okay, so this is important viva questions. You have to know this very important question. Now the parasympathetic system will be acting on there is something else. So what happens in the sympathetic system also, there is some called NN and M1 receptor which will act on which has a preganglionic nerve which forms the acetylcholine, and this acetylcholine in the postganglionic, if it forms the norepinephrine, this is the preganglionic, this is the postganglionic, and it will act on the neuroeffector junction of the smooth muscle. So these are the receptors which are acting on the sympathetic nervous system. These are the receptors for the sympathetic nervous system. Similarly, in the parasympathetic, when it acts on NN receptor and M1 receptor, and the pre is the acetylcholine, the post ganglionic nerve is the effector receptor is the M1, M2, M3, M4 and M5 which acts for the parasympathetic. मतलब अगर देखा जाएगा तो सिंपैथेटिक के लिए अल्फ़ा 1, अल्फ़ा 2, बीटा 1, बीटा 2, बीटा 3, और पैरासिम्पैथेटिक के लिए M1, M2, M3, M4, M5 दोनों के रिसेप्टर्स कहाँ पाए जाते हैं? न्यूरो इफेक्टर जंक्शंस में ऑन द स्मूथ मसल्स. बट इम्पॉर्टेंट चीज़ क्या है कि जब पैरासिम्पैथेटिक का बात होगा देन देयर विल बी ओनली, चाहे वो प्री हो या पोस्ट हो, उसमें एसिटाइलकोलिन रहेगा. जबकि अगर सिंपैथेटिक का बात होगा तो स्टार्टिंग में गैंग्लियन से पहले वहाँ पे तो एसिटाइलकोलिन रहेगा प्री गैंग्लियोनिक में. बस पोस्ट गैंग्लियोनिक जो होता है वो नॉरएपिनेफ्रिन रहता है. क्या रहता है? नॉरएपिनेफ्रिन इन द न्यूरो इफेक्टर जंक्शन और द स्मूथ मसल. बट जहाँ नॉरएपिनेफ्रिन नहीं रहता है वहाँ पे मान लो अगर एसिटाइलकोलिन का बात करते हैं कि प्री में भी एसिटाइलकोलिन, पोस्ट में भी एसिटाइलकोलिन, तो इट विल एक्ट ऑन द M3 रिसेप्टर इन द स्वेट ग्लैंड्स एंड इट इज़ द एपोक्राइन ग्लैंड्स. ओके, और एसिटाइलकोलिन मान लो अगर प्री रहता है प्री गैंग्लियोनिक नर्व में न्यूरोट्रांसमीटर, और पोस्ट गैंग्लियोनिक में डोपामीन रहता है विच विल एक्ट ऑन D1 एंड D2 रिसेप्टर. दिस इज द रीनल वैस्कुलेचर. तो यू शुड नो कि व्हाट इज लाइक पैरासिम्पैथिक नर्वस सिस्टम स्टार्ट कहाँ हो रहा है, नॉट पैरासिम्पैथिक सॉरी, पेरिफेरल न्यूरल सिस्टम में सोमैटिक नर्वस सिस्टम एंड ऑटोनॉमिक नर्वस सिस्टम होता है, राइट. सोमैटिक नर्वस सिस्टम में क्या हो गया? पोस्ट गैंग्लियोनिक नर्व से पाया जाता है विच विल एक्ट ऑन द न्यूरोमस्कुलर जंक्शन ऑन द NM रिसेप्टर. दैट इज द NM, M फॉर मसल, राइट. नाउ इन द ANS में सिंपैथेटिक और पैरासिम्पैथिक दोनों होता है. ठीक है, तो सिंपैथेटिक का एक सिंगल अगर हम बोले तो प्री गैंग्लियोनिक नर्व में अगर NN रिसेप्टर पे काम करेगा एड्रिनल मेडुला में, तो नॉरएपिनेफ्रिन और एपिनेफ्रिन रिलीज़ करेगा, नॉरएपिनेफ्रिन और एपिनेफ्रिन रिलीज़ करेगा. ठीक है, नाउ ये दिस इज़ इम्पॉर्टेंट. नाउ दिस इन पैरासिम्पैथेटिक एंड सिंपैथेटिक there is a common pathway in the neuroeffector junction, that is a smooth muscle or any smooth muscle, any glands ठीक है। तो वहाँ पे क्या दिखेगा तुम्हारा, प्री सिंपैथेटिक में एस्टाइलकोलिन अगर मिलेगा, प्री गैंग्लियोनिक नर्व में अगर एस्टाइलकोलिन एज ए न्यूरोट्रांसमीटर मिल रहा है और पोस्ट में भी तुमको एस्टाइलकोलिन न्यूरोट्रांसमीटर मिल रहा है, देन दिस इज द parasympathetic, okay, and it will act via the M1, M2, M3, M4, M5, and in the sympathetic if there is a pre-acetylcholine, ठीक है, so जो पोस्ट गैंग्लियोनिक नर्व जो होता है उसमें नॉरएपिनेफ्रिन भी हो सकता है, या फिर एस्टाइलकोलिन भी हो सकता है, या फिर डोपामीन भी हो सकता है। तो नॉरएपिनेफ्रिन कहाँ पाया जाता है, तो दैट इज द न्यूरो इफेक्टेड जंक्शन मेनली इन द स्मूथ मसल एंड ग्लैंड्स, एंड इट विल एक्ट वाया द अल्फा 1, अल्फा 2, बीटा 1, बीटा 2 एंड बीटा 3। जबकि अगर देखा जाए कि प्री गैंग्लियोनिक भी नर्व में एस्टाइलकोलिन और पोस्ट गैंग्लियोनिक नर्व में भी एस्टाइलकोलिन, तो यहाँ पे स्वेट ग्लैंड में पाया जाता है, विच इज़ एन एपोक्राइन ग्लैंड, दैट इज एक्ट ऑन द M3 रिसेप्टर, ठीक है। बट इम्पॉर्टेंट क्या है कि M3 रिसेप्टर सिंपैथेटिक एंड पैरासिम्पैथेटिक दोनों में काम करता है, दिस इज इम्पॉर्टेंट, ठीक है। उसके बाद क्या हो गया, दिख रहा है ये M3 में ये सिंपैथेटिक का M3 है ये, और ये पैरासिम्पैथेटिक का M3 है, ठीक है। तो दिस आर यूज्ड फॉर कंपेयर एंड कॉन्ट्रास्ट, okay, and also in renal vasculature what we see in renal vasculature, acetylcholine is the pre-ganglionic nerve neurotransmitter and dopamine is the post-ganglionic nerve neurotransmitter which acts on the D1 and D2 receptor. So this is very very important, this is very very important concept graph, ठीक है। मैं एक बार में भले अच्छे से नहीं समझा पाया तुम लोग को, ठीक है, अपने से इसको बना के इसको टिप ऑफ द टंग हमेशा याद रखना। ये अगर नहीं समझ में आता है, ठीक है, तो तुमको आगे में कुछ भी समझ में दिक्कत होगा, बिकॉज इफ़ यू नो द रिसेप्टर देन यू नो व्हाट इज़ द एगोनिस्ट ऑफ द रिसेप्टर, व्हाट इज़ द एंटागोनिस्ट ऑफ द रिसेप्टर, और कौन कौन सा बीमारी में क्या कंडीशन होता है, किस में क्या बढ़ाना है, किस में क्या घटाना है। तो यू शुड नो द बेसिक एंड देन वी हैव टू मूव अहेड, मतलब पहले बेसमेंट बनाना होता है, उसके बाद बिल्डिंग बनाना होता है। हम लोग बिल्डिंग का टॉप फ्लोर हमेशा नहीं बनाते हैं, ठीक है, कि बैठ के सिर्फ बीमारी पढ़ लिए, उसका रिसेप्टर्स पढ़ लिए और बस एग्जाम देने चले गए, नहीं। फर्स्ट यू नो द रिसेप्टर्स, ठीक है, रिसेप्टर जानने के बाद उसका एगोनिस्ट एंटागोनिस्ट का नाम जानो, वो किस किस ऑर्गन में पाया जाता है, उसके अकॉर्डिंगली जब सिस्टम वाइज तुम लोग कंपेयर करके पढ़ोगे फार्मा एंड पैथोलॉजी, तो वहाँ से तुम लोग देखोगे कि वही सेम रिसेप्टर्स में कुछ प्रॉब्लम्स होते हैं, और कोई भी दवाई हम लोग देते हैं तो वो किसी न किसी रिसेप्टर पे काम करेगा, ठीक है। तो ये सब थोड़ा सा जान के रखो, ठीक है। मे बी आई वॉज नॉट सो फ्लुएंट इन मेकिंग यू अंडरस्टैंड, बट प्लीज़ रिवाइज दिस पर्टिकुलर टॉपिक, दिस इज़ वेरी वेरी इंपॉर्टेंट, ओके, वेरी 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 इंपॉर्टेंट। यहाँ पे फॉर द डिस्कशन, फॉर 3 minutes डिस्कशन, ओके, लेट्स सी, यू सी दिस इज मेडुला ऑब्लोंगेटा, ठीक है, ये कंसेप्ट को तो जानता है हो, ठीक है। दिस इज मिड ब्रेन, पॉन्स, मेडुला, एंड दिस इज द स्पाइनल कॉर्ड, ठीक है। सो दिस इज द मेडुला, दिस इज द एंड पॉइंट ऑफ इट, मेडुला, राइट। नाउ यहाँ पे स्टार्ट होती है दिस क्रेनियल नर्व्स, और फॉर द पैरासिम्पैथेटिक सिस्टम दिस इज द 3rd, 7th, 3, 7, 9, 10, okay, 3, 7, 9 and 10, this is very important. They are the parasympathetic cranial nerves. Now what will happen, it will act on the ciliary ganglion, which will act on the ciliary muscle and circular muscle of the iris, okay. Now this is the 7th nerve, which will act on the sphenopalatine ganglion and submaxillary ganglion, and sphenopalatine will act on the lacrimal gland, submaxillary will act on the submaxillary and sublingual gland, and the 9th, the glossopharyngeal will act on the otic gland, otic ganglion, which will act on the parotid gland, and the 10th will act on a ganglion which will act on the heart, lung and intestine. This is the vagabond nerve, okay. Now from the sacral part, here comes a group of nerves called nervi erigentis, which will act on the pelvic ganglion, and it will act on the kidney, bladder, colon and genitals, okay. This is a very very basic concept, okay. This is a pure anatomy which will be helpful to understand basis of medicine, okay. So this is the first video for the Viva, but let's see how much you get, like from this video.